I'll start now with a little bit of history, pre-Neuropixels. This is how the number of recording sites per shank grew over time before Neuropixels. And what you can notice here is that this is like 1953. There were these electrodes in glass, and they were amazing because they could give you the recording from one neuron in the living brain, which is just incredible. And over the years, people started putting more and more of these recording sites in one shank. And by shank, it's a very strange term. I mean, the thing you put in the brain, you know. If you put 2 electrodes in separately, like my fingers right now, that would be 2 shanks. Okay, and the number of recording sites per shank grew. For example, we got to the tetrode in the late 1980s, and then we got to the 16-channel silicon probe, and then we got to these polytrodes. And you can see what the limitation is on these polytrodes, because basically every recording site, which is a little circle here, is attached to a wire. And by the time you get to the top of the shank, or into the shank, it's all wires. So this has been the limiting factor, such that if you wanted to have more recording sites, you had to have more wires. And Neuropixels breaks this Neuropixels breaks this connection between the number of sites and the number of channels. And in particular, also, Neuropixels does something else. If you look at the sizes of these things, these wires were about 5 microns in the old days. And by the time we were, at the polytrodes, these silicon probes, they were about 1.5 microns. Now, if you go look at around the same time in the 2010s, what chips were like, the wires in there were more like 0.01 microns. And so if we could make wires that are as thin as the ones in these chips, we would have much less of a problem putting these wires in. And so this was the main goal of Neuropixels, was to use, the main method of Neuropixels was to use modern contemporary silicon fabrication techniques to make electrodes. And the width that was obtained is about 0.2 microns. One could go way smaller. I mean, in a modern day mobile phone, wires are way smaller than that. I think Carolina will answer this, but probably a factor of 100 smaller. But then there's issues of heat. And so this is what Neuropixel does. So this is the situation before Neuropixels, and then a dot appears here with 1,000 sites per shank and wires that are 0.2 microns. So this is what a Neuropixels probe looks like. It's got a 1 centimeter shank, and then it's got a chip that is about 1 centimeter in size, and then there's a flex cable, and then there's a head stage and a connector. And what's super cool about it is, among various things, is that what comes out is fully amplified and digitized. So in principle, you don't need a laboratory. You just need to buy this, and in principle, it could go straight into a laptop. In practice, it can't go straight into a laptop because it doesn't speak USB or FireWire or any of these protocols that a laptop would know how to read. And so one needs to go into an intermediate little box that converts the digital to digital so that actually a laptop can read it. But you don't need a lab. When I was starting in neuroscience, it was A, impossible to record from hundreds of sites, and B, once it became possible, it would cost hundreds of thousands of dollars. And now instead, this is a 1,000 euro probe that if you don't break it, that's all you need to spend. If you break it, that's topic for tomorrow. How easy is it to break? And the sites are separated by 20 microns, and so it's possible to record from neurons appearing in multiple sites. And so this is the fabrication method. Folks at Imec, which we'll hear much more about, make these sheets, these wafers, where they produce the silicon. And so you have here this rectangle here appears here, and then every rectangle is attached to the shank, and they produce them. They mass produce them. So this was work that was a collaboration of a number of groups and people, and Mick and I at the time were secondary to all this when they started. And it was a bunch of scientists such as John O'Keefe and Christoph Koch, and a bunch of engineers such as Tim Harris, Barun Datta led the development at Imec. Carolina Mora Lopez led the actual development together with Silke, and she is going to give the next lecture. And so they work at Imec, and as Carolina will explain, Imec is a very unique place. It's a non-profit research semiconductor institution. Usually, from what I understand, they are in charge of ensuring that Moore's law is obeyed. So Moore's law says that the semiconductor field need to grow at a certain speed, and used to be a description of the past, but it was adopted by the semiconductor industry as a guideline for the future. And so various companies such as Intel, etc., know exactly where they need to be in 2 years and 4 years and 6 years and they go to places like Imec to make sure that they can do that. And so the result of this was a paper that you've probably seen, otherwise I doubt that you'd be in this course, but if you haven't seen it, go check it out. It was a paper that came out in 2017 where we described Neuropixels 1.0. Well, at the time it was just called Neuropixels. And I'll just briefly go through these things. Nick, you will go into this stuff in more detail, right? Yeah, okay. So an example of the recordings that you can do with Neuropixels probes is you can insert, for example, 2 probes, one more anterior in a mouse brain, one more posterior. And as you go in, by the way, the probe is 1 centimeter long. The mouse brain is less than 1 centimeter in size. So most of the shank is outside, okay? Or at least a large fraction of it, if not most. And so you'll see, for example, this probe here, probe 2, is going through visual cortex, and it's going through hippocampus, and it's going through thalamus. And this probe here is going through motor cortex, and then it's going through striatum. And this is a raster of the spikes recorded there. And this is hundreds and hundreds of spikes of neurons, which is just, at the time in 2017, was just outrageous, because none of us had ever recorded from this many neurons at the same time. And time goes really fast. It's only 4 years ago. Now it's considered kind of normal, at least by us. I don't know. But at the time it was shocking. And just to give you an idea, this is maybe 800 neurons. And my entire PhD thesis in the 1990s was about maybe 100 neurons. And it took 5 years. Anyway, so you can also use these probes to plan some very interesting penetrations that might go through regions of the brain that you're interested in. This is a figure made by Nick, where he decided that brains can be tilted left and right, which is fine with me, but I wouldn't have. So I wouldn't tilt the animal if I were you, anyway, but you can if you need to. And so, for example, somebody who's interested in the visual system might want to go through primary visual cortex and lateral genicular nucleus. Somebody who's interested in the motor system may want to go through primary motor cortex and caudate putamen. Somebody who's interested in learning and effects of value may want to go through prelimbic cortex and then caudate putamen and then the basolateral amygdala. And you can do all sorts of, plan all sorts of trajectories, and this is going to be the topic of homework 1, which is today. But I also encourage you to look at brain regions that you're not interested in, because the beauty of the probe is that it goes through way more brain regions than you want to, because it's so long, and you end up discovering things. One of the things you might discover is that the particular phenomenon that you're super interested in studying, and you think I have to go to the basolateral amygdala to study it. Yeah, you'll find it there, but you might find it in lots of other places. Okay, and so it allows you to have a broader view of what the brain does. So the probes work well in all sorts of brain regions. Some brain regions are, harder. For example, again, these are different measures of the quality of the ability of the probe to record, how many neurons you're going to get per structure, how many neurons you're going to get per site. And you can see the cerebellum is challenging, because on average, you're only going to get 0.2 neurons per site. And so it's a tough place to be. But otherwise you'll find all sorts of other stats that are good. And in general, every neuron is going to be covered by a bunch of sites, which is useful in spike sorting. The probes have been used for chronic recordings, and you'll hear much more about it in this course. These are data from chronic recordings in an implanted rat. And the probes have been used, once you have these chronic recordings, you can ask, for example, at the same time, how a bunch of cells would have, for example, their grid fields. How do the grid fields of multiple cells at the same time, how do they look, these grid fields, when you have all the cells recorded at the same time, which was in 2017 was an open question. And so you can also do chronic recordings in mice, although as you will see with the newer probes called 2.0, these are even easier in a way, and because the probes are lighter and smaller, but we were able to show that, you can record for, in this case, 153 days, and the total firing rate is decreasing, but not that crazy. And, you get nice waveforms also on the last day. And one of the things that the consortium, when I say we, I mean the consortium, I personally played a very small role in this generation of Neuropixels. One of the things that the consortium struggled with was light sensitivity, because essentially they're called Neuropixels because it's basically the same technology that you would use to make a camera, but if you're sensing here electromagnetic radiation instead of light. And so some of the first generations of Neuropixels that never saw the light of commercialization actually had active sites that amplified things on the site, and those were tremendously light sensitive. And light sensitivity is bad because you don't want your recordings to be influenced by whether a light is flashing in the room, or if you're doing optogenetics, you'd like to not have artifacts when you shine light. And so this is again from the same paper, examples of the fact that if you have any kind of light waveform that includes a sharp transient, you will end up with a little artifact in the spike band, the actual potential band. Neuropixels, as Nick will explain, have recording two bands: the LFP band, which is lower frequency, the local field potential, and the AP band, action potential band for spikes. Now, I should point out that these curves are obtained with extremely high light intensity. So if you use lower light intensities, even these waveforms with transients are going to be okay. And also the transients don't saturate the amplifier, so you don't have to worry about that. They will look weird when you go spike sorting, so you might be okay. But if you really don't want transients, you may want to use something like a raised cosine. And so as a couple of people, and especially Maxime Beau on day 3 will explain, it is totally possible to do optogenetics while using the Neuropixels probes. Here's an example of a recording where there's channel rhodopsin in layer 4 excitatory neurons. These are all the spikes recorded as a function of depth. And as you can see, when they shine light, a bunch of neurons were activated. And you can do the opposite by activating inhibitory neurons with light, and you see that the cortex shuts down, but not the hippocampus. And you can see here 2 examples of waveforms, right, that were used. Here is a slow ramp, like a raised cosine, and here's a faster set of oscillations, kind of in the gamma. Okay, all right, so in the gamma range. Nick, will you tell me if I get out of time? I think I'm still okay with that, but
will do.
How much time do I have?
You have 7 more minutes.
7 more minutes, okay, moving on. So a huge chunk of this course will be devoted to what you do from when you have the data to actually inferring the activity of individual neurons. And so a lot of time will be spent talking about KiloSort, which is the software we use to go from raw traces to identifying the spiking of individual neurons, and to manual curation, data processing, which we do with a program called Phi. And this will be the subject of homework 2. which will be assigned tomorrow after Nick goes very much in detail about how you do spike sorting. And well, Marius Cacciatario will go in the details of spike sorting, and Nick will go in the details of manual curation. Of course, it really helps to have ground truth. Occasionally this ground truth is available. So some recordings were made in this, which I don't think ever became a paper, in which they were doing intracellular recordings at the same time as recording with probes, both Neuropixels probes and earlier probes called NeuroSeeker, which had some issues and never saw commercialization, but they were very cool. And so there are these data sets, if you're interested in spike sorting data that has a ground truth. Nick, will you talk more about the ground truth data?
During the spike sorting section, probably, yes.
Okay, cool. So another very important thing is once you have planned the trajectory and obtained your recording is to understand where your probe actually went. And so what we do is align our probes to the Allen Common Coordinate Framework. By the way, this is all for mouse, and I believe now the similar tools are starting to be available for rat and for maybe for other animals too. And so the Allen Common Coordinate Framework is a volume of mouse that has been averaged over lots of mice and annotated every 10 microns. And we use that, and Matteo will show a tool. Matteo works for the International Brain Laboratory. She will show a tool that we use to align after we've done the anatomy, which will be described by Steve West. She'll talk about how we decide where exactly a channel was along a shank, along the track. And so this tool will give you data that looks like this, well, she'll give a much better lecture than this. Now, Nick will also tell us about the kind of science you can do with these probes and the fact that you can insert multiple probes in multiple mice and get a very broad view of how some processes occur in the brain. So in the case of Nick's as a postdoc, he recorded from 10 mice in 39 sessions while the mice were doing a particular behavior. And this was, in total, there were 92 probe insertions. He recorded from lots of places around the brain. And I'm not going to say anything about his results, but I just want to point out that this is a new kind of science that you can do. Say you want to study what happens when the mouse is thirsty, which is something that was done in the Dijsselhoff lab using similar techniques. Well, you can record all over the brain and answer and find out what the activity of neurons is when the mouse is thirsty. Or say that you want to know what happens when a mouse hears the sound of another mouse, which is not an experiment that anybody has done, as far as I know, yet with Neuropixels. But you could go throughout the brain and answer that question. And you might discover that some crazy regions that you didn't even know about, for example, I didn't know about zona incerta, which is here, where we have 220 neurons. It turns out to be the most interesting region where we found, correlates of this behavior where mouse turn a wheel. And had we stuck to the old style of neuroscience and just gone to the usual suspects for our lab, we would not have seen that. Usual suspects in terms of regions. So this was Neuropixels 1.0. They were released, I think, around 2019, I think. But the research went from 2012 to 2018. And it's acquiring more and more citations. This last bar is an extrapolation based on present citations, which makes me think that a lot of labs are adopting it. In fact, I hear from Imec that around 400 something labs are using it, which is a large number because not that many labs record from the brain. So what's happening next? Next is Neuropixels 2.0, which is a project that started in 2017 and should end next year. And you might have seen this paper, which came out in Science earlier this year. And here, Nick and I played a much bigger role than in the, well, Nick greatly helped write the first paper, but he definitely wrote the second paper. And it's a collaboration that has many of the same people. Carolina is there, and many others. Tim is still there, etc. But also has new collaborators and a new set of funders, which is in some ways similar to the previous one, but it also includes new ones. And so what does Neuropixels 2.0 do? Well, as a first step, it minimizes the size of the electronics, which makes it easier to put multiple probes next to each other and makes it easier to put them chronically because they weigh less. And then, as in addition, we also added the possibility of having 4 shanks instead of 1 shank. So now you have 5,000 recording sites. One thing I didn't explain, by the way, let me step back to the original Neuropixels 1.0. You have basically 1,000 sites, okay? But you need to choose 384 of them to record from. This is how we break the connection, you know, the link between sites and channels. Channels, which are essentially the wires that come out, can be reassigned fairly rapidly to different sites. But that means you cannot record from all 1,000 sites at the same time, which in mouse is not a huge problem because these 1,000 sites anyway, you have a hard time putting them all in the brain because it's over 1 centimeter. In a rat or a bigger animal, you might be sad that you can't record from all 1,000 at the same time. Okay, so in Neuropixels 2.0, you can choose to have a very similar probe with slightly more sites and 1 shank, or a probe with 4 shanks. And you may wonder, why would I want the 1 shank? Why shouldn't I just want the 4 shanks? Well, the 4 shanks is probably going to be a bit more expensive. They'll be commercialized in 2022. So you'll be a little sadder if you break it. But also, it's definitely a little bit harder to put it in the brain. So if you do a durotomy, remove the dura, it's fine. But if you don't do a durotomy, it is definitely easier to insert 1 shank than 4s. We use various methods to sharpen the probes, and you'll hear that about that in this course. And now, also very likely, from what I understand, is that we will have a version that has 4 shanks, 5,000 sites, and you're able to record from 4 times as many channels. So you could decide that you record from an entire shank or from other combinations. And so this is still upcoming. It hasn't appeared yet. And I can't promise that it will, but I strongly hope that it will. So what kind of science can you do with these probes? Well, for example, you can do things like insert all 4 shanks inside the striatum and record these waves of activity. I'm now showing figures from the science paper that came out earlier this year. And so, or you could decide that you put 2 4-shank probes in, and you alternate which sites you record from. So they're not all recorded at the same time, but you can do, you know, you can record from 6,000 sites in a mouse brain, which has never been done. And I was particularly delighted that it actually was more than Elon Musk's implant. Of course, these records are made and broken faster than I speak, so we shouldn't have too much glee over that. But one of the things that we're super proud of is the chronic stability of these probes. We inserted them for a long time in mice and rats all over in 6 labs, and we found very good stability over time. So this is over, I'm sorry, the x-axis got cropped, but it's days since implantation. And you see that many of these lines are sloping down. So you have, you know, fewer neurons over time and a little bit less firing rate over time, but it's a very gentle decay. And so we have very good stability, and, you know, the amplitude of the spikes in particular doesn't seem to go down. If you had a lot of damage with gliosis, you would see the spikes shrink and shrink and shrink until they disappear. All right, so another thing that was really advantageous here, and here the advances are both in hardware and in software, is the ability to undo the movement of the brain. So it turns out that if you just record from the brain of a mouse, and you don't do, and the mouse is not doing anything particular. In this case, I don't even think the mouse is able to run, but Nick, correct me if I'm wrong. You'll find that this is a representation that we use in which dark means large spikes and light means small spikes. You'll find that things, and this is the length of the shank, you'll find that, well, a portion of the shank, you find that the brain moves relative to the probe. And so that it would be very hard to spike sort and then during this period, because the spike you're trying to detect is now in a different section of the probe, which is, by the way, still a good problem to have, because if you had traditional electrodes, the spike would be gone. Here, a nice thing is it has moved to a different portion of the probe. It's not entirely gone, but it makes it hard to write papers, because if whatever your mouse is doing causes the brain to move, and you're finding the neuron to come and go, it could be because the brain is moving, not because the neuron is coming and going in its firing, right? So what Nick did in this collaboration, still the same paper, is he made the probe move up and down on purpose. Okay, and I believe Nick will explain this better. So he imposed an actual motion of the probe. And by the way, this section here, highlighted here, is the section shown here. And when he imposed that motion, of course, there was a lot of motion in the recorded spikes. But the beauty of that is that now this is ground truth, because we know how the probe is moving. And using this software called Kilosort 2.5 and exploiting the fact that in Neuropixels 2.0, this is a very simple change, but the recording sites are closer to each other and they are aligned on a line, which means that as the brain moves, most of the movement is going to be perpendicular to the probe. This actually depends on how you inserted the probe, but anyway, it's going to be easier to see the spikes go up and down. We were able to use this software to undo the motion, and you can see that it undid both the motion that happened before that we didn't control and the motion that we imposed. And it actually estimates the motion, and it ends up estimating something similar to this curve, which I'm not showing. Okay, the very coolest thing, though, I think, was done by Anna Lebedeva and Michael Okun, and Anna will give a lecture on the third day, which was to follow the same neurons over days. And here I'm just summarizing her results. Anna used the trick in which she was recording from primary visual cortex, and she used the trick for labeling individual neurons by seeing whether they respond to certain images. Okay, and this gave a signature because different neurons would respond to different images. So this is images by neuron. And then she compared across days where the neurons had the same signature, and she found that the best fitting neuron, called also a unit, Carolina, do you have a question? No. She found that the best...
Matteo, let me just say 3 minutes left.
Okay, thank you. The best fitting neuron for each neuron is the same neuron. In other words, on day 14 and day 15, she was largely responding to the same, she was largely recording from the same neuron. And many of the neurons remained stable over, months, but in some cases, in some recordings, the number greatly decreased. Now, this was a chronic implant that did not move. At the end, you're not able to remove the probes. You've wasted €1,000 because the probes are glued in. But you will hear from Yo Isugai various implants that now allow to remove the probes and reuse them. Okay, I got a couple more slides very briefly in case you're interested in where things are going in the future. One direction that we're trying to go into is Neuropixels opto, which is a combination of electrical recordings and light emission. And the idea is that right now, if you want to stimulate with light to do optogenetics, you need to put in a probe like an optic fiber that is very large and will do lots of damage. And if you want to record at the same time, you need to put another probe, an electrical probe in. And that is very invasive, and they're not necessarily matched in space, etc. So we would like to make a probe that does both and emit light at 2 different wavelengths and record. And we have funding from Wellcome and from Allen Institute to create some prototypes. Here's a picture of a prototype. It works. We have some evidence that it also works in a brain, but these projects are extremely expensive. Neuropixels 1.0 and 2.0 in combination was about €6 million just to Imec. And so we need to find a similar amount of money now to make opto happen. And finally, I won't go into details, A very exciting development is these Neuropixels NHP for non-human primates, which means that they're a little stiffer and longer than the other ones, which can be put in non-human primates. And the amazing thing is that they have actually been inserted in humans recently. This is a study that came out in bioRxiv a few months ago, and I don't really need to go into it, except that they sterilized the probes, they put them in various ways inside the brains of human patients in the surgical room, and they obtained some very nice recordings, although in most humans they had a hard time grounding the probes and there were lots of artifacts. So clearly this is a field that still needs to move. And the biggest challenge is the fact that the brain of a human has tremendous movement relative to the probe. The probe is attached externally, or if you wanted to do chronic recordings, which has never been done in humans, you would attach it to the skull, and this would be a problem. Because there's massive movement, which is in the order of 100 microns, okay? And which they tried to undo in this study by having video tracking of the brain movement and trying to spike sort after alignment. And after they did that, they obtained some nice looking units, but this is a challenge. All right, I'm done. I imagine you have lots of questions, but the whole course will cover what I just went the topics I covered. And so, feel free to ask questions, but I suspect many of the questions you have will be answered if you just stick around for the next 3 days, because I just gave an overview of what all of these people will talk about in detail, okay? And just as a reminder, please use the Zoom Q&A if you want to ask about the lectures, use the Zoom chat if you want to ask more general questions, and if you are among the interactive participants and you have questions about doing the homework, you can use Slack. But anything more general, please put it on Zoom. Okay, so thank you so much. I'm going to stop sharing now, and let's see how we're doing. I don't want to start by being late and being the first person who's late, so Carolina, if you want to start sharing your screen, please do. And in the
Sure. Can you hear me well?
Yes.
Great.
Great. If there's anything burning in the Q&A, where can we find out more about NHP neuropixel development? Nick, will you talk about it? Carolina, will you say anything about Neuropixels NHP?
I don't have anything, but I can comment on something during the presentation.
Okay, so I will ask Barun Datta at Imec if he wants to send us a slide or something, and we will make sure we show it by day 3.